And so our next presenter is going to be Dr. Kathleen Boese-Battaglia from University of Pennsylvania, and she's a professor of biochemistry and assistant dean for academic initiatives. One of the amazing things about choroideremia is that at a fundamental level, although we sort of know what happens, we still don't know exactly at a molecular level what happens. And Dr. Boese-Battaglia is looking into those things, and her research focuses on the intricate relationship between metabolism, diet, disease progression, and particularly how lipids or fats contribute to health and disease. And with that, Dr. Boese-Battaglia, thank you for being here.
So I don't know if I want to see the audience for my slides. Thank you very much to the organizers for the very generous invitation. And thank you to everybody who I have met. I am absolutely humbled to be standing up here, and I appreciate all the CHMers and your candor and your discussing what everyday life is like, because it certainly puts context into what we're doing. So I'm going to talk a little bit kind of different from what we've heard today. Dr. Mack, so to speak, gave a wonderful overview of the relationship between photoreceptors and RPE, and I'm hoping to expand on that. So we're all very familiar with this notion of you are what you eat. There are endless studies on how our regular diet alters metabolism, it alters various cellular functions. There's things like fatty liver, there are predispositions to cancer, etc. But a further assessment of that really looks at how 3 macronutrients, so sugars or carbohydrates, amino acids or proteins, and fats really modulate tissues at a cellular level. So what happens in a very confined space, for our purposes, this confined space is that interconnection, that ecosystem that we can call the photoreceptors and the RPE. And while we do have metabolomic data, which I'm happy to speak to anybody about, I'm going to focus today's talk on what we see as real changes in lipid composition. So again, this is, thank you, Dr. Mack. We are all, oh, can you see my pointer? Okay. We are all familiar with the photoreceptor cell layer and then the RPE cell layer, and they form this very intimate, tight link with each other. Now, it's very important when we think about how these things function is that these cells cannot make more. They cannot be replaced. So in contrast to skin, where if I have a cut, my skin wound heals, etc. We are born with more or less as many photoreceptors and RPE cells that we will have. So understanding how these 2 layers interact and understanding how the RPE layer, the cell layer that is involved in the degeneration in most cases of CHM function is incredibly important. So one way of looking at this is to ask ourselves, how are lipids handled in the RPE? So there is an intercellular pool of fats. They come from both the munching off, I love that term, Dr. Mack, of the photoreceptors. They come from de novo synthesis. Diets high in sugars, high in fructose, are going to increase lipid synthesis. They also come from plasma lipids. They are also removed. They provide fuel and building blocks to the photoreceptors, and excess lipid is very often removed and taken out through the bloodstream. What's important for all of us to know is that REP1 modified proteins are involved in many of these pathways that balance lipids. Okay, so in order to try to understand how CHM mutations alter lipid composition, so we can, ultimately target some of these lipid metabolic pathways for therapeutics, we need to know what the various lipid species are like. So We rely on several model systems. The first of these is actually a series of cells. These are samples that Jean Bennett collected years ago. They are from skin or blood samples from patients who have choroideremia. So again, for anyone in the room or anyone you know who has donated, thank you so much. We are able to take those cells and really grow them on a plate, stick them in an incubator, and add different ingredients, because we can think about this as kind of a cake, and have those cells differentiate or become RPE cells. So we now have a model where we can look at how RPE cells from CHMers may or may not be similar to unaffected healthy adults. Okay, and one of the first things we looked at was actually lipid accumulation. So the more green you see here, the more lipid, the more fat that is actually accumulating in the cells from patients with CHM. So then, okay, that tells us, yeah, there's lots of fat there, or some fat there. What we really want to know, and this actually goes back to a question that Dr. Mack was asked previously, is what is the composition of that fat? What does it look like? And if I know what it looks like, if I know what its origin is, maybe I can target various pathways that those REP1 associated proteins affect. So we do what's called lipidomics, which is a large-scale study of lipids. So just very briefly, we use those RPE cells that we've derived. We then do a series of isolations. We add some nice standards to make sure what we're looking at is what we think we're looking at. We then separate them on a system that differentiates them based on size. So each type of lipid, so we were talking this morning about DHA 22.6, there's thousands of other lipids, has a distinct pattern or a distinct fingerprint, so we can determine what your lipid profile looks like from those RPE cells. We do a series of data processing, and then we compare different data sets from both patients to, in my case, what we did is compare to animal models and also compare to human plasma that was isolated and published from patients with CHM. Okay, so let me just walk you through this because I think this is important for a number of reasons. When you look at this whole lipid profile, so you have, let's just hypothetically say 100 lipids, and you separate it based on where they're coming from. You see a whole bunch of little dots. Each dot represents a run or an analysis. And what you'll notice is that all of from a certain patient cluster together. So here is one CHM, here is another, and they are distinct from the healthy individuals. Now, normally as a biochemist, I would look at this and go, oh my God, they are all over the place, okay? But what was really fascinating to us is there were two incredibly common themes that carried through, even though these look wide apart. And they are that there is, and don't worry about actually reading the detail, but there are differences in the type of lipid. So how you read a picture like this, it's called a heat map. So those compounds that are more abundant or at a higher level are going to be a lighter yellow color. Those compounds that are a lower number or a lower value are going to be darker. Okay, so you see CHM is a whole series of compounds on this side, and what we're calling wild type or unaffected individuals is on this side. And I draw your attention to the arrows here. where we see, and this is going to be important, a substantial increase in a lipid called ceramide. Okay, there's a lot known. Feel free to Google. It's a lipid that, promotes cell death. We also see a substantial decrease, and this goes back to a question this morning, in the polyunsaturated fatty acid DHA or 22,6. So clearly, we now have two distinct changes in our sample populations. They are an increase in ceramide lipids and a decrease in omega-3 lipids. Now, we then went a little further because really what we want to do is target various pathways for therapy. And the in vitro system provides some information, but an animal model, as was just explained, either the pig or we use a mouse, gives us a completely intact system where you can look at diet, you can look at inflammation, because increased lipid deposits increase inflammation, and you can look at the retina and the RPE altogether. So we did the exact same type of lipidomic analysis that I talked about before in mice. Again, the choroideremia model mice separate out very nicely from the wild-type mice, and we see exactly the same trend. So we see less DHA, or 22,6, omega-3 fatty acid, in the choroideremia mice, and we see an increase in ceramide. Okay, so again, an increase in ceramide lipids and a decrease in those omega-3 lipids. Now, first of all, for those of us who work in this, this in and of itself was huge in some ways because it's not always common to have human cells, so the patient-derived RPE cells and a mouse model match so nicely, so that was one real positive. But what was even better is we then took our data and did a series of statistical analyses and compared it to data from plasma isolated from patients with CHM. And this is a paper that was published now about 5 years ago and found, to our actually surprise, that ceramide was increased not only in our in vitro model, it was increased in the mouse, but it was also increased in the plasma of patients with choroideremia. And we also found that DHA, so 22,6, was decreased again across the board in mice, in the patient-derived cells, and in the plasma of patients. So one idea that I kind of throw out there is, can any of this be used as a biomarker potentially with more research of progression or predisposition? So that's kind of one thought there. So given I only thought I had 10 minutes and I have a lot to say, I'm just going to summarize very quickly. Our patient-derived IRP cell culture system showed deposition of lipids, and it also showed increases in ceramide, known to induce cell death, and decreases in omega-3 fatty acids. But I want to leave us with a little bit of what's going to happen next. So we now have the systems where we can, both in short term in our culture system and a mouse model, start asking, how can we decrease these lipid deposits? You know, we don't want a lot of lipid building up. A lot of lipid tends to induce inflammation, so we have several compounds we're looking at. One is LGM-2605. It's a flaxseed derivative, so for all the nutritionists who say flaxseed is good, flaxseed probably is really good. It's a potent antioxidant. We have another compound called SS38 that increases your ability to use those fatty acids for energy. which means you're going to be less reliant on sugars, which is actually also very much nutritionally positive. What about ceramide? So there are several current drugs on the market, and those goes kind of back to what was said this morning. Statins will decrease ceramide, metformin decreases ceramide, and fenofibrate decreases ceramide. So all of those, again, are approved for other things, but they are out there for us to try. How do we attack this decreased omega-3 fatty acid? Obviously, one of the best ways is the omega-3 fatty acid supplements. I agree with Dr. Mack, actually. I take one in the morning and I take one at night. I take the one at night because your ability to use DHA and those polyunsaturated fatty acids actually is circadian, so you use as much of it at night as you use during the day. So if you double dose, you can't take too much. I mean, I don't think anyone overdoses on omega-3, but you kind of get a double boost. So that was just kind of an aside. But our next steps are to then take those compounds and to do targeted compound screens. So this allows us to take our cell cultures and ask if any of those compounds I've listed are going to help balance the fats within the RPE, and then we can take those compounds, translate it into looking at our mouse model, and ask about things like efficacy, safety, etc. So with that, there's just 2 references for individuals who may be interested in detail. There's a number of drugs that look at ceramide, and we've also published on that flaxseed derivative LGM-2605, both of which decrease toxic lipids. So I just want to acknowledge the people in my lab who did this work. Anu is a talented senior investigator, and Alina Armarco are students who really like the idea of grinding through all this lipid data because they could do it from their beach house. I'm serious, that's what they did. But they got it done, and they were terrific. And again, I thank the Choroideremia Foundation for funding and the $1 Million Bike Ride. And with that, I know we're taking questions at the end, so thank you for your attention.
As a cardiothoracic surgeon, I see the effects of lipids all the time. And Dr. Mack, I give statins all the time too, for the same reason, not necessarily with eyes. I'm also grateful because Dr. Boese-Battaglia very ably and nobly serves on our scientific advisory board. And I'm grateful for all she does to help us decide what to fund and what grants are good enough that meet our members' expectations to fund.